Hi, it's Aubrey Garner, and today I'm going to talk about the cell cycle. I'll discuss how it is regulated and what happens if the cycle loses control. First, I'll give you an overview of what the cell cycle is and the different phases that make it up. Then I'll discuss how the cell cycle is regulated through its internal timing mechanisms and checkpoints, and then how the checkpoints work through the use of proteins such as M-phase promoting factors and platelet-derived growth factors. Lastly, I'll talk about how the loss of these controls can lead to cancer. All cells are made from pre-existing cells. The cell cycle demonstrates the events that occur in a cell's life from the time it divided from its parent cell to the time it divides itself. The first part of the cell cycle is interphase. This is the phase in which cells spend most of their time. Interphase is then divided into three subphases: G1, S, and G2. During the G1 first gap phase, the cell grows. During the S synthesis phase, the cell copies its chromosomes. And during the G2 second gap phase, the cell grows even more as it prepares to divide. When the cell is ready to divide, it then enters the mitotic M phase, which is broken up into two parts: mitosis and cytokinesis. Mitosis refers to the splitting of the nucleus, and cytokinesis refers to the splitting of the cytoplasm, but oftentimes they are both just called mitosis. Mitosis is where prophase, metaphase, anaphase, and telophase occur, and then cytokinesis occurs afterwards. When a cell does not need to divide anymore, or if it's a cell that only divides once in its whole life, like nerve cells or certain muscle cells, they will enter the G0 phase. This is like a waiting area, and if they need to start dividing again, they can re-enter the cell cycle. How do cells know when to go from one phase to the next, or when to start dividing? Well, the timing of the cell cycle is controlled by several factors. Scientists discovered that if a cell in the G1 phase and a cell in the S phase are fused together, the G1 cell will jump ahead to the S phase. This suggests that there are signaling molecules in the cytoplasm that help control the timing of the cell cycle. This is evidence for the cell cycle control system, which is a set of molecules in the cell that both start and coordinate major events in the cell cycle. Think about the cell cycle control system like a dial on an old washing machine. Once you start the washer, the timing device keeps it going until it hits a stopping point, like the second rinse option, for example. The cell cycle control system also continues on its own due to its built-in clock. But it also has stopping points that are presented by internal and external factors. Some internal factors are checkpoints. Checkpoints are points in the cell cycle where the cell is either told to stop or to keep on going. These checkpoints make sure everything that should have happened in the cell cycle up to that point has happened correctly before the go-ahead signal is given off. The G1 checkpoint is important because if a cell gets the go-ahead here, it usually proceeds in the cycle and divides. But if it gets a stop signal, then it will go into the G0 phase. The G2 checkpoint determines whether the cell is ready to start mitosis, and the M checkpoint makes sure that all of the sister chromatids are correctly attached to the spindle, microtubules, before they start separating. All of these checkpoints reveal how highly regulated the cell cycle is because the cell has to pass each step before it can move on to the next. This also lessens the chance of any mistakes occurring throughout the cycle. As I said earlier, that the cell cycle control system is controlled by molecules. Well, what are these molecules exactly? Fluctuations in the amount and activity of two types of proteins called kinases and cyclins control the timing of the cell cycle. Kinases are enzymes that activate or inactivate other proteins. They provide the go-ahead signals at the G1 and G2 checkpoints. Most of the time, kinases are in an inactive form and stay at a constant concentration throughout the cell cycle. To be active, the kinases have to attach to a cyclin, so they are given the name cyclin-dependent kinases, or CDKs for short. Cyclins accumulate during the S and G2 phases, and as the number of cyclins goes up, so does the number of CDKs. One type of cyclin-CDK complex is the M-phase promoting factor, or MPF. As the name implies, MPF is the go-ahead signal for the G2 checkpoint that determines whether the cell is ready for mitosis. This graph shows that as the cyclins accumulate, or the cyclin concentration increases, the amount of MPF activity increases. This makes sense because as the cyclins accumulate in the G2 phase, their increase tells the cell that it is almost time to start mitosis, and the amount of MPFs increase, which tells the cell whether or not it can start mitosis. And at the peaks, mitosis occurs, and the cyclins drop off dramatically because they were used up during mitosis, and the MPFs are recycled for the next time they are needed. This diagram helps you see better the processes that are going on. As you can see, the cyclins accumulate in the S and G2 phases. The cyclin-dependent kinases attach to the cyclins to form MPFs. This gives the go-ahead signal to start mitosis. The cyclins start to degrade because they are no longer needed. The cyclins degrade and their concentration goes down until the cell gets to the S phase again in its next cycle. The remaining CDKs are recycled until they are needed again. So far, I have talked about internal factors that influence the cell cycle, but there are also several external factors. An example of an external factor would be that some cells require a growth factor. A growth factor is a protein that is released that tells surrounding cells to start dividing in order to pass a checkpoint. For example, platelet-derived growth factor, or PDGF, is released when you get a cut or wound. This signal tells the cells surrounding the wound to start dividing in order to replace the damaged cells and to heal the area. Two more external factors are density-dependent inhibition and anchorage dependence. Density-dependent inhibition is where crowded cells stop dividing because there is no more room for the new cells, since cells only grow in a single layer. If two cells are touching, a cell surface protein sends out a signal to both cells telling them to stop dividing, even in the presence of a growth factor. Anchorage dependence describes how cells must have something to attach to in order to divide, such as a petri dish. Cancer cells usually divide well past a single layer, forming a clump of overlapping cells. Cancer cells do not follow density-dependent inhibition or anchorage dependence. Also, cancer cells do not listen to normal cell signals that regulate the cell cycle. Therefore, they can divide excessively and even invade other tissues. They also do not stop dividing even when growth factors are depleted, which suggests that they might not need them in order to divide. Normal cells will divide about 20 to 50 times in culture before they stop dividing, age, and die. But cancer cells can divide indefinitely if they are given a continual supply of nutrients. The process that converts a normal cell to a cancerous cell is called transformation. The immune system usually recognizes the abnormal cell and destroys it, but if the cell is not destroyed, it can proliferate and form a tumor, which is a mass of abnormal cells. If the cells stay where they originated, then the tumor is benign. But if the cells move to another organ or organs, then the tumor is malignant. The spreading of these cancerous cells to different locations is called metastasis. Benign tumors can often be removed with surgery, but malignant tumors require chemotherapy treatment to destroy the cells. However, chemotherapy also harms some good cells, which is why you often see hair loss and sickness in cancer patients. In summary, internal and external factors control the timing of the cell cycle, and loss of these controls can lead to cancer, and I hope that's helpful.